Now we will be talking about your non-fermenter gram-negative bacilli. The general characteristics is that they are non-fermenters, so they are oxidizers. They break down carbohydrates oxidatively. And for non-oxidizers or asaccharolytic, they are not able to break down carbohydrates either fermentatively or oxidatively. So, oxidatively metabolize carbohydrates and they require molecular oxygen as the final hydrogen acceptor. Note that 15% of all gram-negative bacilli isolates is a part of this. Septicemia, meningitis, osteomyelitis, and wound infections are clinically related to your non-fermenters. The risk factors for your clinical infections is that for immunosuppressed, trauma, foreign body implantation, and infused fluids. There are three tests that are commonly performed to facilitate your identification. You have your oxidase, your growth on your McConkey agar plate, and the glucose oxidation in your OF medium. We now talk about your Pseudomonas and your Burkholderia species. So here on top is your Pseudomonas, and here on the bottom is your Burkholderia species. Pseudomonas species are gram-negative bacilli or Coccobacillus. They are motile with polar or polar tufts of flagella except for your Burkholderia malae. So they are oxidase and catalase positive. They grow on your MAC. And there is a good growth at 42 degrees of incubation. Also oxidizes your carbohydrates. First up, we have our Pseudomonas aeruginosa. This is the causative agent of lupus or your Bacillus pyocyanus, and it is one of the most common. You have two pigments, you have your pyocyanin and your pyoverdin or your fluorescein. Pseudomonas aeruginosa produces a grape-like odor or a corn or a tortilla-like odor. This is because of your amino acetophenone. Okay, so you have your in MAC, they appear to be blue-green. In BAC, they produce beta-hemolytic and they have green metallic sheen. And in your cetrimide agar, this is used as a selective media. Virulence factor for your Pseudomonas aeruginosa is its fimbriae and pili used for attachment, your exotoxin A, which inhibits protein synthesis, you have your hemolysins, which destroys the cells, and your alginate, which resists phagocytosis. This genera is an opportunistic pathogen, so in normal individuals, they disrupt protection provided by epidermis. So usually it is uh, acquired through nosocomial and ecthyma gangrenosum, your skin infection. Okay. Examples of your Pseudomonas species are Pseudomonas fluorescens, putida, and your Stottzeri. So these three species have no virulence factors. Your Pseudomonas fluorescens is seen in UTI and in wound infection. They are usually an environment contaminant, so comes from your food spoilage and your plant infections. While for your Pseudomonas putida, it is seen in your septicemia, infections of arms and legs, and it can be grown through your hospital sinks, so that's where you can collect them at times. This is pyoverdin positive. For your gelatin hydrolysis test, your Pseudomonas fluorescens will turn out to be positive, and your Pseudomonas putida will turn out to be a negative result. Now move on to the next species, which is your Pseudomonas stottzeri. The one that you have to remember here is that this is a soil denitrifier. So your colonies are dry and wrinkled, and they appear to be light brown in color. Next up, we have your Burkholderia malae. This is the causative agent of your glanders disease. the infectious disease in horse through ingestion of contaminated food or water. So this is also known to be a potential agent for biological warfare. In humans, the disease can occur in four basic forms. You have your acute localized infection, septicemic illness, your acute pulmonary infection, or your chronic cutaneous infection. Burkholderia malae is the common isolate. So a direct contact with contaminated food or medical solutions or disinfectants and respiratory equipment is the mode of transmission. So have your Burkholderia pseudomalae. So this is an agent of melioidosis, which is also called as your Whitmore disease or your Nightcliff-Gardiner's disease. So it is very similar with Glender's disease. It can also be a potential agent for biological warfare and biological terrorism. Okay. The bacteria is found in contaminated water and soil and is spread to humans and animals through direct contact as well. For the lab diagnosis, we use gram staining. And in your gram stain, you can see a medium-sized rod. So you have your Burkholderia diminuta, which appears to have a long rod structure. You have your Burkholderia malae, which shows a Coccobacillus shape. And your Pseudomonas pseudomalae, this appears to be a bipolar stained bacteria. So in the gram stain, you can see it as a safety pin appearance. You have to memorize this class, the three different kinds of species and their descriptions. So all grow in routine culture media and in McConkey agar, except your Burkholderia vesicularis. Your PC agar or OFPBL, your oxidative fermentative base polymyxin B bacitracin lactose. This one is used for your Burkholderia cepacia.